All right, so this is the last lecture on lipids. We've covered the saponifiable lipids. Remember, they had these esters here linked to glycerol. We talked about the sphingolipids. This is not an ester, this is an amide, right? Fatty acid is in an amide linkage to sphingosine. There is an ester linkage here to the phosphate in the alcohol of sphingosine, the phosphate in the alcohol of this amino alcohol, and then here we have the glycosidic bond. Lastly, now we're going to talk about steroids, and then also we'll talk a little bit about cell membranes. Okay, the steroid nucleus. You do not have to memorize. We have, two. These are not aromatic rings. They're not benzene rings. They're just cyclohexane rings bonded, fused together here, and then we have a cyclopentane here. You do not have to memorize how this looks. Just as I talked about, maybe you remember this guy. 1, 2, 3, 4, 5, 6, 7, 8, 9. There were some double bonds in here. Remember that this one that wrapped around itself, this was arachidonic acid. It's 20 carbons. It was arachidonic acid. Arachidonic acid. So it was the only one that looked like that wrapped around itself. And then we also had the derivative of the arachidonic acid that formed this. I kind of messed that up. Just do it like this. That cyclopentane ring, this was the prostaglandins. Again, you don't have to memorize the where the alcohol groups or anything like that. There was an alcohol group here and here, but we had that cyclooxygenase. Remember that oxygenated it and made this cyclo part, this cyclopentane ring. So you just need to recognize the basic structure. Likewise here, this is the steroid nucleus. These steroid hormones, these are all derivatives of cholesterol. So you'll see that just a moment here. All right, so again, here we have cholesterol. We have the steroid nucleus, these 4 fused rings. There's 3 cyclohexanes and then a cyclopentane. And then it has this alkyl chain. Okay. Again, you don't have to actually memorize it. It's got 1, 2, 3, 4, 5, 6 carbons and a couple branches on it. You don't have to memorize it. There's these methyl groups here. One thing you should recognize, however, this is an alcohol. All right? Alcohols and an OL. The only functional group here is the alcohol, and therefore cholesterol is an OL. It's an alcohol. They say here it's the most abundant steroid in the body. In and of itself, it is not a hormone. You think of steroids, you think of hormones. It's not a hormone all by itself. However, these steroid hormones are all, of course, derivatives of cholesterol. One last thing I should mention about cholesterol. This little bit of polarity here is not really enough to make this amphipathic. So when I talked about fatty acids, we can actually just talk about this real quick. There's something called a fatty acid micelle. If you go back, I can't remember what chapter that was, but we talked about the fatty acid, right? So it has a long hydrophobic tail, and then it has the acid group, right, which has a negative charge. So the fatty acids form this thing called a micelle, and all the polar head groups, finish this here, are on the outside, right? Because they're on polar and they're going to interact with water. Right? So this part here again is polar. The interior is hydrophobic. I'll just write nonpolar. Okay? And the way soap works is dirt itself is nonpolar, so it gets trapped inside the center of this micelle, and then with all the polar groups on the outside, it gets washed away. Right? So you need an amphipathic molecule that can do that. One that has a polar head group that can react with water, and one with a hydrophobic tail that can react with the dirt that goes inside. All right. This is not like that. This is not enough to make it amphipathic. So it's just simply very hydrophobic, not soluble in water. In the diet, we get cholesterol from meats, milk, eggs. If you happen to eat brain tissue, I don't know. I'm not saying you're a zombie, but you know, some people eat brains. I don't know, not me. But remember, brains were high in sphingolipids and glycosphingolipids. So they're also high in nervous tissue is also high in cholesterol. It doesn't mention that here. Maybe you'll see that in the next slide. But anyway, you don't need cholesterol in your diet. You don't need it at all because we can make our own. We make our own in the liver. Okay, well, here it is. It's needed for cell membranes, right? Brain and nervous tissue. So it's found in nervous tissue. Most people don't eat brains, but you know, if you eat, I don't know, calf brains or something, there's a lot of cholesterol in there. Also, remember I mentioned it's hydrophobic. So it's nonpolar, hydrophobic, and it clogs the arteries and forms the atherosclerotic plaque. So this is what a normal artery should look like, where the blood can flow through. This is almost completely occluded, so there's nothing getting through there. The pipe is jammed shut, and your drano is not going to work here. You're going to have to consult not a plumber, what do you call this? A cardiologist to fix this. So just as I was saying, it's made in the liver. You don't need it in your diet. We can make plenty on our own. You can get it from foods, right? And foods that are high, right, in it, liver, eggs, lobster, surprising. Fried chickens, all these fatty things. They don't have brains on here. But what you're looking for is a total cholesterol less than 200. But then when they look at your cholesterol levels, they want to look at the ratio of LDL to HDL. And you want a lot of the HDL and a little bit of the LDL. We can talk more about that in another lecture. Bile salts. Two things you need to know about bile salts. These are derived from cholesterol. And they're not steroids, but they are derived from cholesterol. So they're synthesized in the liver from cholesterol. They're stored in your gallbladder. So the gallbladder is nothing more than it's storage sac essentially. So it's a little muscular bag that stores these bile salts, and essentially, they have a polar region and a non-polar region. These are amphipathic. Right. So what does that mean? Well, they emulsify fat particles. This is in your food, I should say in your stomach, or small intestine actually, secreted into the small intestine. So they're secreted into the small intestine, and basically they act as soaps, right? They act as soaps, and they help emulsify or solubilize, if you wish. If you remember, well, we didn't do this lab, but if we had a test tube, let me just draw a test tube, right? And we put down here some water in a test tube, and we put some oil in here. Okay, the oil is less dense, it would sit on top. Okay, well, so it forms two layers. Well, we could, what I want to do here is just explain what we mean by emulsify. Okay, we're going to have the same thing. We're going to have the oil, we're going to have the water, but in addition to that, we'll add some soap. Some soap or some surfactant, a detergent, a surface active agent. And basically what this does is it makes it one layer. So okay, the water is this pink stuff. We have oil mixed in. We don't get two layers anymore. We have one layer, and it's the soap that does this, that emulsifies them, and basically just brings together what is an emulsion, it brings together into one mixture, one homogeneous mixture, two things that normally wouldn't mix. Right? Well, this is important because the fats in our diet, they're not soluble in water, but our digestive tract is full of water, right? So we need to emulsify these to provide a large surface area, and they'll be broken down or digested better. Right? So that's basically what these bile salts do. Let's take a look at what they actually look like. Now again, you don't need to know the structure or like memorize the structure. I'm not going to move an alcohol group on you and say, oh, you got it wrong. But recognize the 4 rings. Okay, well, we got a cholesterol nucleus here, got this long thing here. But now look at this part. This is something you should recognize. So this is something you should recognize. This says this is a polar region. This is a salt. And what they've actually done is put glycine on this. So this is a glycolate, sodium glycolate. It's a sodium salt of glycolic acid. So glycine is another one of these amino acids. So here's the acid. This is the alpha carbon. Here's the amine. So this part here is glycine, and it's an amide bond to this. There was an acid here as well, but this is this polar region, right? This is a salt, and so this is the polar. This interacts with water, so it's hydrophilic. This is the nonpolar region, right? This is hydrophobic. If you have a molecule that has both hydrophilic and hydrophobic regions, right, it acts like a soap, and it's amphipathic. Lipoproteins, as I mentioned before, lipoproteins, LDL, LDL, we have HDL. We'll talk more about proteins later, but let's just kind of briefly talk about these lipoproteins. So they're kind of like, really not like a soap micelle, like a fatty acid micelle, like a soap, like the soap globs or globules that I had mentioned earlier, but similar to that. So now, so they've got, they're looking at here is the surface, and you've got a protein, right? So this is a protein. Inside you have cholesterol esters. What is that? Well, remember cholesterol had this. I won't draw the whole molecule, but I'll draw enough of it. Right? You had this alcohol here. Well, you could esterify a fatty acid to that alcohol. Yeah, so we have a fatty acid attached to cholesterol. That's a cholesterol ester. Triacylglycerols. Right? This is one way that triacylglycerols are moved through the body, through the blood. They get incorporated into these lipoproteins. The surface here are made up of glycerophospholipids. So remember glycerophospholipids? It just says here phospholipids. So you had the glycerol backbone. Let's just do it this way. You had 2 fatty acids coming off. Fatty acid 1. Fatty acid 2, and then here you had the phosphate and the amino alcohol. This was the polar part. Well, notice the polar part is on the outside. This is what interacts with the water. The inside is the hydrophobic part. Right? The inside, just like in the soap globule and that soap micelle, was a hydrophobic part. Hydrophobic. And we also have, well, here they actually show a cholesterol ester. So here's a cholesterol bonded to a fatty acids. This here is cholesterol ester. That's this guy. But notice we also have cholesterol in the membrane. And of course the alcohol group is going to be on the outside. We'll talk more about this in just a moment, but not only in the membrane of these, or the exterior of these lipoproteins, but also in regular cell membranes you have cholesterol. So cholesterol has a number of different roles. Okay, we'll summarize those later. but one of them here is in cell membranes. So I talked about LDL and HDL. HDL and LDL. We differentiate them based upon their density, and that's actually how they're isolated, is by differences in their density, high density or low density. Really what's more important, at least as far as the cell is concerned, is the nature of these proteins. LDLs have one particular type of protein, HDLs have a different type of protein, and that has a big difference in how these lipoproteins function. We can talk more about that later, but I just say they have different proteins on their surface. All right. Something that you might be familiar with, well, actually, I expect you to be familiar with this, right? I just drew that test tube, right? Oil is less dense than water, so we had water down here, and then the lipids, the oil was on top, right? So it's less dense. So these lipoproteins, there's I talked about high density, low density. There's also very low density. They actually give the density. Just don't worry about the density. Just recognize very low is less than low. And then there's these things called kilomicrons, which actually have the lowest. Now, what I want you to focus on are, you can really just focus on the top line and the bottom line. Okay? The ones that are the lowest density have the highest amount of lipids. So here for LDLs, it doesn't change as much, but maybe you want to look here at the cholesterol, phospholipids, right? So the kilomicrons, VLDL, LDL, what we're looking at here is the lipid amount is going up. So I'll say amount of lipid increases as the density decreases, right? Density is getting low, so this is lower density. So lower density basically means higher amount of lipids. Conversely, down here, the protein is increasing. Right, those are those surface proteins, right? So protein increases, density increases. So the higher the protein, the higher the density. So recognize these two trends. Recognize that kilomicrons are the lowest. And not only are LDLs and HDLs are probably the two most important, but they're not just differentiated based upon their densities, they're also differentiated by the proteins that happen to be present in each. They simply have different proteins in them. Here is where those proteins make a difference. These proteins that I was talking about. Okay, so again, I'm just gonna focus on here the LDLs and the HDLs. Let's look at the LDLs first. Okay, so you eat some food, some lobster, some eggs, something that has a lot of fat in it. So these dietary lipids, right? They go into your small intestine, the gallbladder, muscular sac squeezes down, sends in those bile salts that are also derivatives of steroids. They go into first kilomicrons, get metabolized through. This is actually, we'll talk about this after 22 or 23. We talk about how these lipids are metabolized. It's something called beta oxidation, happens in the mitochondria of cells, and we can get energy from them. If we have excess, what happens? Well, it can go back up to the liver, right? And it can be either then stored. The VLDLs can take them and store them as in fat cells at adipocytes, or they can be excreted, and they can be used as bile salts. But notice the HDL basically takes the excess and brings it to the liver. And then the liver can get rid of them. So the HDL move cholesterol to the liver and essentially, well, I'm going to put it here, there's more room. HDLs take lipids out of the blood. And so what they do, remember these lipids carry cholesterol esters. So the HDLs lower blood cholesterol because they remove the lipids from the blood. So they take lipids out of the blood and they lower blood cholesterol, which is good because you don't want the cholesterol in your blood forming plaques and atherosclerosis and whatnot. And so that's why they call this the good cholesterol. Cholesterol is cholesterol, but the HDLs lower blood cholesterol, so it's kind of a misnomer. They call it the good cholesterol. LDLs, on the other hand, they take lipids and cholesterol that is formed in the liver. Where does this go? It goes into the bloodstream. So LDLs carry cholesterol to cells and deposit excess cholesterol in arteries. So what do LDLs do? LDLs increase blood cholesterol. So this is termed the bad cholesterol. You don't have to know. I mentioned what really, well, let me just briefly mention this. So HDLs and LDLs have different proteins on them. The proteins that the LDLs have on them are receptors or bind with receptors in peripheral tissues, right, to peripheral cells. So they're taken from the liver to the blood to the peripheral cells. HDLs don't have that. Instead, they have proteins on them that bind to receptors in the liver, right, in the hepatocytes in the liver cells. So they take the excess out of the blood, take it to the liver, which can then get excreted. Steroid hormones, I'm not going to talk a lot about this. The only thing I expect you to know is recognize a steroid nucleus when you see it, these four rings fused together. You should recognize steroid hormones come in a couple different types. There's the sex hormones. So the sex hormones, androgen, so testosterone in males and estrogen in females, and they're responsible for secondary sex characteristics and gamete production, ovulation. And then we have adrenal corticosteroids. So adrenal corticosteroids, these are produced in the adrenal glands, and they actually have a lot of different functions. But the adrenal glands, why are they called adrenal glands? Well, they're located, let me use red here, they're located on the top of each kidney. So they're adjacent to the kidneys, or the kidneys perform renal functions, so they're adjacent to renal. So adrenal, that's where adrenal comes from. It's adjacent to the renal. And these regulate a bunch of things, including fluid balance, electrolyte and fluid balance, inflammation response. And I include here cortisol and cortisone. And these are interconvertible. And the only thing I really wanted to focus here, essentially they're cortisol, cortisone, essentially the same thing. But cortisol ends in OL. It has an alcohol here. Cortisone ends in ONE. It's a ketone. Right? We go from the alcohol to the ketone. Right? This is an oxidation. Oxidation. Going back the other way from the ketone to the alcohol, this is a reduction. So there's an enzyme called reductase, 11B. This is 11 carbon, so 11 beta reductase. It reduces this carbon 11 here from the ketone to the alcohol. The other enzyme is a dehydrogenase. Remember, oxidation, one of the definitions we had for oxidation was a loss of hydrogen, right? So what does a dehydrogenase do? It removes hydrogen, right? It removes this hydrogen, goes from the alcohol to the ketone. Some more, well, again, here's cortisone, aldosterone. You don't have to know the structures of these, or even be able to tell the difference between an adrenal corticosteroid, a sex hormone like estrogen or testosterone. That's not important, at least not for this class. Anabolic steroids, these are synthetic steroids that are used for performance-enhancing drugs. They have a number of really bad side effects, sleep disorders, liver damage. Because they're derivatives of testosterone, if you're getting your testosterone from these exogenously, your testes don't have to make them, so it does decrease sperm production and testicle size and hypogonadism. So not really a good thing to be taking. And also it says here hair growth. Also, you get acne. So if women take these, they start getting male characteristics, and I mean they may have uses in terms of gender reassignment and that sort of thing. But as a general rule, these are something to stay away from. Quick learning check here if you want to pause it and test yourself, see how much you remember from all this. Identify each of these as 1 through 4. Fatty acid, whatnot. Okay. Cholesterol. Cholesterol is a steroid. It's the most abundant steroid, although it's not really a steroid in the sense that it's not a hormone, but it's the most abundant steroid in the body. Now, next we have glycerol, 2 fatty acids, a phosphate, and a choline. This is phosphatidylcholine. Right? This is a phospholipid. Right? So I'm actually going to put it here. Right? So it's a phospholipid. It's not a triacylglycerol because there's only 2 fatty acids. Glycerol tristearate, that is a triacylglycerol. That's 3. Sphingosine. So we have sphingosine, a fatty acid, phosphate, and choline. This is myelin, right? This is that stuff that forms a myelin sheath. This is phosphosphingolipid. It is, not only is it a phospholipid, it's also a sphingolipid. So actually 4 and 5 because it contains a phosphate and it contains sphingosine. Estradiol, well, that's a steroid. Bile salts, also steroid. Lipids in the plasma membrane. All right, well, there's a couple things here. It could, well, primarily it's 4, right? The phospholipids, remember the phospholipids had the polar head group that contained the phosphate and amino alcohol, and then 2 fatty acids. But we also have steroids, and you'll see this in just a moment here. It could also be 2 because cholesterol is also found in the cell membrane. So as I mentioned, we'll talk about cell membranes just briefly here. So this here's the outside of the cell, here's the inside of the cell. Note the thing that's going on here is a lipid bilayer. Bi means 2, so we have 2 layers of lipids, right? We have an outer layer and an inner layer. The inside here, we see all those little hydrophobic tails. That's a hydrophobic region. Hydrophobic, that's made up of those hydrophobic fatty acid tails. The polar head groups are the balls on the outside, right? Made up of the, we can do it here, right? Fatty acid 1, fatty acid 2. Here we had the phosphate and the amino alcohol that make up the polar head group. That's these little balls here. So we have the outside of the cell. This is a water environment, so we need the polar head group on the outside. But inside the cell is also, the cytoplasm is also full of water, so we also have water on the inside of the cell. Okay? And then we have proteins embedded in the membrane. Some are transmembrane proteins that go all the way through. Some have a channel in them to shuttle sodium ions in or glucose in, that kind of thing. Some are receptors, so they are just sitting on the outside. And then we also have carbohydrates. If you remember the blood groups that we talked about, one of the uses for carbohydrates was in cell signaling or recognition. And so a lot of proteins are glycosylated. So we have a glycoprotein, the carbohydrate decorating the surface of protein. We also have them bonding to sphingosine, where we had those gangliosides, right? So we had a bunch of carbohydrates attached to the polar head group of sphingolipid. So yeah, that's what they're showing here, glycolipid, glycoproteins. And lastly, they don't show it here, these little yellow balls, right, these guys here, these yellow ones, that's cholesterol in our model. I think on the next slide they'll identify that. No, not the next slide, but next to last slide. Okay, so this fluid mosaic model. Basic fluid, what they mean is the molecules can move around. Molecules can move around. It is not, the fluid membrane is not a rigid surface, but instead those proteins that you saw, they can kind of swim around in that lipid bilayer. So that's the fluid part. The mosaic means it's made up of a bunch of different parts. So a mosaic is a I just say composed of many parts, right? We have the lipid, well, here's it, the lipid bilayer, and it says here it contains proteins, carbohydrates, cholesterol. That's the mosaic part. The membrane is fluid-like rather than rigid, right? And that's what I was just talking about. The proteins can move around on the surface and they can communicate with one another, and they can move around. They're not locked in place. This was the same slide as before, and you can, yeah, so here we say, right, we have cholesterol in there. So the cholesterol actually, when we talk about the fluid mosaic model, the cholesterol regulates membrane fluidity. So the amount of cholesterol in the membrane isn't fixed. It can change, and as it changes, the membrane can either become more or less fluid, and that's actually really important. Take a upper level like cell biology course or something like that. You'll learn more about the role of cholesterol in membrane fluidity. Membrane fluidity. So is it membrane really fluid and allows these proteins to move around freely, or is it more rigid? That's the role that cholesterol plays. All right. The end.